All right, I think we should get started, although I've been informed by the organizers that we should get started because otherwise every panel will start to become late. There's still some people who will be trickling in from the big panel that's going on right now with Varoufakis and Stiglitz and others. So let's get started with this afternoon's session. Vexed and intriguing question: inequality and behavior. Inequality and claims about genes. At least one speaker today is going to be speaking about genes, which is Professor Daniel Benjamin. Anyway, my name is Arjun Jayadev, and I'm Associate Professor of Economics at UMass Boston, and I work with INET as well. Normally, we would have had 3 speakers, but unfortunately, Professor Derloff could not be here due to some unavoidable circumstances. So we will have only 2 speakers here. The silver lining is that they can actually get through their presentations, which typically is not always possible in an INET conference. All right, so just to introduce them. We'll have first Professor Daniel Benjamin, who's an associate professor in the economics department at Cornell and a visiting associate professor at USC. He's one of the world's leading young researchers in the fertile intersection between 2 related fields, behavioral economics and genoeconomics, which is the incorporating genetic information into social science kind of understandings. He's also the co-organizer of the Social Science Grant Genetic Association Consortium, which is a collaborative research infrastructure trying to bring in genetics into social science research. So he'll go first and speak for about half an hour, and then after that we will have Professor Michael Gazaniga, who's the director of the Sage Center of the Study of the Mind. He's one of the world's leading researchers in cognitive neuroscience, which is the study of the neural basis of mind, and he's written a number of books which are sort of classics in the field, including The Integrated Mind, The Social Brain, The Mind's Past, and most recently, Tales from Both Sides of the Brain. So he's also a member, by the way, of the President's Council on Bioethics and a member of the Academy of Arts and Sciences. So what we'll have is both of them speak for half an hour, and then we'll have probably half an hour open for questions. So, Professor Ben.
Thanks. So I'm going to, so my talk is called The Fourth Law of Behavior Genetics, which I'll explain shortly. But I'm going to start with the story of how I got into this area of research that my collaborators and I called genoeconomics, at the intersection of genetics and economics. And then, since the name of the session is Claims About Genes, I'm going to talk about some true claims, as well as some common misconceptions about the role of genes. And I should acknowledge at the outset that this work is highly collaborative and multidisciplinary. There's more collaborators than I can list on one slide. They come from many disciplines: economics, psychology, sociology, genetics, statistics, epidemiology, medicine. And so, the things I'm going to talk about are, very many people have contributed. Now, the starting point for thinking about genetics and behavior is classical twin and family and adoption studies that have been going on for over 100 years now, whose goal is to infer a parameter called heritability, which refers to the percent of variance across individuals that can be statistically attributed to genetic factors taken as a whole. Now, each of the methods for estimating heritability has been criticized, but based on different assumptions, different methods have generally converged on similar conclusions. And so I think at this point we can be pretty confident in the conclusion that virtually all psychological traits have heritabilities in the range of 20 to 80%. And economic outcomes, to the extent that they've been studied, seem to be similar, maybe somewhat lower in terms of their heritability. Now, what these heritability estimates imply, I think a true claim, is that it means that there exist genetic variants that, taken collectively, explain the percent of variance that we call heritability. So, for example, for educational attainment, years of schooling has been estimated to have a heritability about 40%, means that there should exist genetic variants that if you take them together, they should add up to explaining about 40% of the variance across people. So it should be possible to find them. Now, when I started getting interested in this area, before graduate school, it was around 2001, right after the Human Genome Project was completed, I was coming at it from the point of view of a behavioral economist, which is my training. And so I was interested in finding genes with big effects that would matter through neurotransmitter systems, other known biological systems. And by studying genes that mattered that way, it would contribute toward understanding the biological mechanisms underlying decision-making. Well, at that time, there was just no data available for studying genetics and behavior. So I spent a few years reading textbooks and doing some twin research. And then finally in 2004, I got access to some data from Iceland. And I followed the research design, which was state-of-the-art at the time, called a candidate gene study, which is where you identify specific genetic variants that are believed to be involved in certain biological systems, and you measure those particular genetic variants and study how they relate to the traits of interest. So I did this with a number of collaborators. We actually found some genetic variants that seemed to be associated with educational attainment. And then, before publishing the paper, we decided we should confirm that the results hold up in other samples. So we tried to replicate, including with this was in a sample of about 2,000 Icelandic individuals. We did a replication study with another 1,500 Icelandic individuals from the same population, also 2 other European descent populations. And to make a long story short, the result did not replicate in any of the other 3 samples. So based on that experience, so this was now 4 or 5 years later, my collaborators and I began to worry that how big of a problem this might be, this non-replication. So we wrote a paper on genetic association studies with cognitive ability. So we looked at a comprehensive review of reported candidate gene associations, and we studied a sample of 3 data sets that collectively had a sample size of about 10,000 individuals, so much larger than the samples in the original studies that reported associations with cognitive ability. And we studied 12 genetic variants that were available across these 10,000 individuals, and tried to replicate what had been these published associations. So this is the result of that paper, the main result. What you see across the bottom here are the 12 genetic variants that we studied. The y-axis here is in units of IQ points. So a standard deviation corresponds to 15 IQ points. So plus or minus 1 IQ point is quite a small effect, much smaller than what had been reported in the original studies. And what I'm showing here is 95% confidence intervals from our 10,000 individuals, so pooling across the 3 samples we studied. And the fact that the confidence intervals all include 0 indicates that there was no statistically significant effect for any of these 12 variants. And the 95% confidence intervals rule out effect sizes larger than 1 IQ point. So at least in this area, it seems to be, this non-replication, non-robustness seems to be a problem. And it's now becoming widely recognized that it's a problem more generally. So you don't just have to take my word for it. What I've put here is a quote from a new editorial policy adopted in 2012 by Behavior Genetics, which is a leading field journal in this area. This is an editorial policy that now imposes very strict standards for publication of candidate gene studies, requiring, for example, independent replication of any reported finding. The editorial policy begins with a statement that I've put up here. I'll just read you the beginning. It says, "The literature on candidate gene associations is full of reports that have not stood up to rigorous replication. This is the case both for straightforward main effects and for candidate gene-by-environment interactions. As a result, the psychiatric and behavior genetics literature has become confusing, and it now seems likely that many of the published findings of the last decade are wrong or misleading and have not contributed to real advances in knowledge." And then it goes on to talk about possible reasons for that. The same policy has been adopted by some other journals, for example, Psychological Science, which is a leading journal in psychology. So this raises a puzzle. There's two facts that, on their face, may seem hard to reconcile with each other. The first is that there's moderate heritability of many behavioral traits. And on the second fact is that there's been a general failure to identify robust, replicable associations with specific genetic variants. So the fourth law, title of the talk, is a proposal that my co-authors and I have for a resolution to this puzzle. We call it the fourth law in the spirit of Turkheimer's three laws of behavior genetics, which are meant as empirical regularities, the first of which is that all human behavioral traits are heritable, and then there's some other summaries of the evidence from the twin and family studies. The fourth law that we propose is that a human behavioral trait is associated with many genetic variants, each of which accounts for a very small percentage of the behavioral variability. Now, there's a sense in which it was kind of known already that you can't talk about a gene for some behavioral trait. But the point here is not just that individual genetic variants have a very small effect, but they have a very, very, very small effect. So small that the samples that have been used to try to identify them to date haven't been big enough. So this is something which I call the power problem. So to give you, why do I think that the effect sizes of the effects of individual variants are small? Well, the existing studies in the candidate gene literature have used sample sizes of between 50 to around 3,000 people. So like 50 in the case of laboratory experiments, up to maybe 3,000 in studies that use larger social surveys. Now, if you use a sample of that kind of size, and maybe the 1,000 range, there's an implicit assumption, if you think that you're conducting a well-powered study, that you have you have an effect size that can explain 2% or more of the variance. And there's reasons to think that's probably not true. So one is that there have been very large genome-wide association studies now in the last couple of years with up to 60,000 people looking for associations with cognitive performance, cognitive ability, and personality traits, and they haven't found much of anything. If there had been traits, if there had been genetic variants that could explain 2% of the variance, they would have had plenty of power to detect them. They would have found all such genetic variants. So that suggests they don't exist. Another source of evidence for what kind of effect sizes might be reasonable to expect is to look at the evidence from the physical and medical literatures where we can be very confident that true associations have been identified because they've been found and replicated in very large data sets. For smoking, one of the strongest association that's been found has an R squared of 0.5%. For BMI, the strongest association has explanatory power of 0.33%. And for height, which is an interesting case because it's now been studied in samples of 250,000 people, and there's over 700 genetic variants that have been found for it, the largest of them accounts for 0.4% of the variance. Now, it's possible that behavioral traits have larger effects than for these kinds of medical traits, but I would think, if anything, it's more likely to be the opposite, that these are traits with more direct biological connections between the genes and the outcomes, and so probably the effect sizes we should expect to be smaller for behavioral traits. And if that's true, then the existing studies have been dramatically underpowered, which implies a pretty high likelihood of a false positive. So to make that point clearer, I think it's worth doing a simple calculation. To simplify, suppose that a genetic variant only had two possible versions. We can call it the high and the low allele of the genetic variant, and we're studying some outcome that has a normal distribution. And suppose we know that there's only two possible states of the world. Either there's a true relationship or there isn't. And suppose we know that if there is a true relationship, the size of the effect corresponds to an R squared of 0.02 of a percentage point. So that number actually comes from somewhere. I'll come back to that later. That is now our best guess about what is a realistic kind of effect size. So what would be the sample size for 80% power in that case? So think what you would guess you would need for 80% power. The answer is about 40,000 individuals. So about 10 times, more than 10 times larger than the largest candidate gene studies that have been done. And now we can ask another kind of question. This is a Bayesian question. Let's say that you find an association that's statistically significant at the 5% level, or you read a paper that reports an association significant at the 5% level. What's the likelihood that that's a true association? Well, you can calculate that probability using Bayes' rule. This gives the answer for several different sample sizes. So this first column is a sample size of 100, corresponding to a laboratory study, sample size of 10,000, which would be like a very large social survey. And this would be a sample of 100,000. And then what I have here in the rows are different prior probabilities. I think 10% would be a very high prior to have on a particular genetic variant, and 0.1% might be more appropriate for a kind of whole genome scan. And focusing on this column, say for a sample size of 100, what you can see is that even if you start with a very optimistic prior of 10%, a 5% significance threshold, finding a significant result, doesn't give you much confidence at all that the result is actually true. In fact, your belief should barely move at all, and you should still think there's a 90% chance that it's not true. The intuition for this is that with a sample size of 100, your power is just a little bit more than 5%. So the fact that you found a statistically significant result means that under the null hypothesis, there was a 5% chance that you would have found what you did. The fact that your power is 5% plus epsilon means that under the alternative hypothesis, there's a 5% plus epsilon chance that you would have found what you did. So having found what you did actually doesn't give you much evidence that that the alternative hypothesis is more likely than the null hypothesis. Now, even with a sample size of 100,000, where your power is virtually 100%, even if you start with a strong prior of 10%, finding a statistically significant result at the 5% level still gives you about a 30% chance of a false positive. And that's because even with power of 100%, finding a statistically significant result only gives you 20 to 1 evidence in favor of there being a real effect. And so this is our intuitions are misleading in this world of very, very small effects. What you really need in a world of very small effects to get convincing evidence is the combination of a large sample size and a very stringent significance threshold. That's how you get strong enough evidence. So I'm going to add to the list of facts a third fact, which I'm going to demonstrate now, which is that with enormous sample, well, this is an implication. If the fourth law is true, it should be the case that if you have very large sample sizes and stringent significance thresholds and you find something, then you should be able to identify robust genetic variants. So with this idea in mind, my colleagues, David Cesarini, Phil Kollinger, and I, in 2011, formed the Social Science Genetic Association Consortium. So the idea was to follow the model of the medical genetics world, which has tried to create very large sample sizes by combining results across many, many data sets. The problem is that no one data set is actually big enough to give you sufficient statistical power. But if you conduct genetic association studies in 50 data sets and then meta-analyze the results, then you can get large enough samples. So this is actually, extremely organizationally complex. It requires a lot of coordination among many, many, many collaborators from all of these data sets. It's typically a very expensive kind of research to do, whereas we've been operating on a shoestring. If we have like 1% of the funding for the kind of medical consortia that do this sort of work. So if we would not complain if INET were interested in contributing some help in our direction. But we've, despite operating on a shoestring budget, I think we've been able to demonstrate that this model can work. So in 2013, we published the result of our proof of concept project, which was a study of educational attainment in a combined sample of about 125,000 individuals. We then followed it up with another paper that explored the robustness of the finding by trying to replicate it in another data set. And to summarize, this picture summarizes the results. What we found in the science paper were 3 genetic variants that were statistically significantly associated with educational attainment at the what's called the genome-wide significance threshold, which is a statistical significance threshold of 5 times 10 to the negative 8th. So it corresponds to the standard significance threshold of 5%, Bonferroni corrected for about 1 million independent units of genetic variation. And what you see here, these are the 3 genetic variants. The y-axis is the effect on years of schooling. The dark bars show what we found in our original sample size, which was 100,000 individuals. And so we found effects, these are 95% confidence intervals. We found effects of about 0.1, so that's 0.1 years. That corresponds to about 1 month of schooling. These bars here, the kind of medium shade, these are the replication sample that we included in the original paper. This was a sample of 25,000 individuals, and all 3 of the significant genetic variants replicated with roughly the same effect sizes. And then the light bars here show the results from our follow-up paper, which was in a sample of 35,000 people, independent of the other samples. As you can see, the effects replicated yet again with similar effect sizes. So, but what are the effect sizes in terms of R squared? The answer is 0.02% for each of these genetic variants. That's more than 10 times smaller than what we're seeing for the complex physical and medical traits that I showed you on the earlier slide. And that's where that number comes from before that I used for the power calculation. So on the one hand, this is good news, which is, I think we now have a research strategy we know that can work for identifying these variants. On the other hand, it means we need to realign our expectations of what can be done with this kind of data. So it's not going to be the case that we can do what I thought we'd be able to do when I first got into this area, which is find genes with big effects that would directly themselves tell us a lot about biological mechanisms. We'll have to learn about functional pathways and overlap with other traits using statistical evidence from many of these genetic variants taken together. So let me now tell you about a little bit of work in progress that's not published yet that I think reinforces this conclusion. We have a new project which we call Educational Attainment 2.0. This is now kind of a redo of the same analysis, but now with a sample 3 times as big. So now we have 300,000 individuals. And what we find now is 83 genetic variants that are associated at this 5 times 10 to the negative 8th significance threshold. We can look at the distribution of effect sizes across these 83 genetic variants. And this is what it looks like. They're ordered now from biggest to smallest. This is 0.1, the effect size we saw in the earlier paper. So we see just not many variants that seem to have bigger effects than that. Almost all of them are smaller, as you might have expected. If we look at the effects in terms of R squared, this is what it looks like. So this threshold here is the 0.02 of a percentage point that we saw in the previous paper. So again, not many seem to have bigger than that. Almost all are smaller. And combined, they add up to about 1% R squared. Now, one thing I want to mention, because it's important for potential uses of these results in social science and economics, is that many of the uses that you might imagine for genetic data don't require having individual genetic variants. They just require that collectively you have a set that are sufficiently predictive. So for example, one use of a sufficiently predictive genetic variable would be, which you could create as an index of many genetic variants, is as a control variable. Suppose what you wanted to do was conduct a randomized evaluation of an intervention designed to improve educational outcomes. Typically, these kinds of interventions, like providing free preschool to disadvantaged families, are very expensive interventions. So anything you can do to increase statistical power is very valuable. And if you have a variable that can explain 10% of the variance, a genetic variable, that you can control for, then what that will do is absorb variance in the residual and improve your precision to estimate the causal effect of the intervention. The fact that you have a randomized intervention means that you're identifying the effect off of the randomization. So you're really just improving statistical power by including the control. So I think that's something that will become actually a very common application in the future if we can create these predictive genetic variables, and we can create these kinds of variables. There's a method for doing so called constructing polygenic scores, which basically uses the estimated effects as weights for creating an index of genetic variants. When we create such an index from our sample of 100,000 individuals, we can construct a polygenic score that explains about 2.5% of the variance across people out of sample. In our sample of 300,000 people, we don't know, the final results aren't in yet, but it's looking like it might be something like 5% of the variance that we can explain out of sample. It's going to get bigger and bigger as we get larger samples, at least up until, it won't be able to exceed the heritability, but it'll get bigger than 5% in the future. And that's already big enough to begin to do some useful analyses, like, for example, using the genetic polygenic score as a mediating variable or studying gene-by-environment interactions. Okay. So I've tried to emphasize what the heritability of behavioral traits implies, namely that there exist genetic variants that can be identified and that they will have a joint predictive power that could be as high as the heritability. Let me end with some things that the heritability of these traits does not imply. That is, some influential misconceptions about genetics. One major fallacy that seems to be made repeatedly over the years, even by experts in the area, is that higher heritability is often misinterpreted as meaning that there's less variance attributable to environmental factors. So, people act as if the heritability plus the proportion of variance explainable by the environment has to add up to 100%. So some trait that's more heritable is therefore less environmental. And that's not true. In fact, in general, heritabilities are going to add up to more than 100% because there's overlapping variance. There's variables that are both genetic, count as part of the genetic variance and also as part of the environmental variance. And that's because genetic effects can be, and in the case of behavioral traits, probably generally will be, mediated by environmental factors. So to give an example, suppose there are genes that matter for curiosity. These genes cause people to go out and read books, and reading books leads these people to be smarter and end up getting more education. Well, those genes involved in this channel are going to be part of the heritability of educational attainment. But if you then look at how much of the variance in educational attainment is explained by reading books, the same channel will show up as part of that variance. And so the sum of those different components will add up to more than 100%. So that's one major fallacy. The other major fallacy is that high heritability is often misinterpreted as meaning that there's little scope for policy to matter. That's also a mistake. Here's a concrete example of this mistake. Paul Taubman was one of the first economists to study heritability. He found that income was heritable about 40%. He published this finding in 1976. Hans Eysenck, a very influential and very smart psychologist, was quoted as saying that these results tell us that the Royal Commission on the Distribution of Income and Wealth might as well pack up. There's nothing they can do about income inequality because income is 40% heritable. So that's a mistake because what heritability says is how much of the variance can be explained in a particular population given the distribution of causes that are naturally occurring. It doesn't mean that if you come along with a new cause, you can't have a big effect that's kind of outside of the variance that's normally explained in the population currently. So to make this point very concretely, Arthur Goldberger wrote a response to the Eysenck quote. He said, "A powerful intellect was at work. In the same vein, if it were shown that a large proportion of the variance in eyesight were due to genetic causes, then the Royal Commission on the Distribution of Eyeglasses might as well pack up. And if it were shown that most of the variation in rainfall is due to natural causes, then the Royal Commission on the Distribution of Umbrellas could pack up too." The same caveat applies to polygenic scores or the variance explained by individual SNPs. We used to keep in mind that high heritabilities and the possibility of finding SNPs that explain variation in the outcomes does not mean that there's no role for policy. Thanks.
Perfectly on time. So, Professor Galvin.
That was terrific. Actually, the title of my presentation is Neuroscience Perspectives on Inequality. And I'm glad I didn't attempt a heritability and genetic analysis because I certainly would have been calling upon the wrong pool of data. I'm going to try and cover some areas of interest that are ongoing in neuroscience and hopefully uncover some ideas in that survey. I just wanted to make a quick note at the beginning that the marriage of two professional fields is difficult. About 6 years ago, the MacArthur Foundation asked me to marry law and neuroscience. I don't know how many of you are familiar with the legal mind, but it's quite different than the scientific mind. And it took 6 years to get the conversation going because of different attitudes, styles of thought, what people thought were important. But now it's going and it's very, very lively. So getting getting the conversation going between experimental science and these large social issues that economists deal with, like inequality, will take a patience, but I think it will pay off. So what do I do to change that thing?
The green button.
The green button. It's always the green button. So, so here's a cartoon that nicely picks up on what we just heard. In the 50s, right after from Watson and Crick on, there was a very simple view. There were genes that determined function, which determined proteins, which determined functions, and there was some low-hanging fruit in that area that people thought that if you had a certain kind of genetic disposition, you were doomed to have a certain way of life, a certain behavior, a certain disease. That's all changed, and today we are now aware that in fact there's all kinds of forces at work in between the fetus, the young baby, the child, and up into, of course, adulthood. And all of that sweep of things, as represented in this cartoon, is very much adding to variation in the final product, and the actual brain that is the structure where all of these genetic influences play out to produce behavior. And it adds to variability, the epigenetic forces that are apparent in the development of the child, the psychosocial forces in our lives leading to variation. And counteracting that in many ways is this incredible resilience that we see in some people, which now has a genetic story, now has an experimental psychological story to explain why no matter what you throw at people, they don't seem to be affected in the way that we commonly think people are affected with such adversity. So you get this increasing variation, and there's just this little cartoon slide just summarizes a tremendous amount of individual papers and efforts where people are contributing knowledge at these various points along the way. And also represented here, we now know that the fetal brain is under all kinds of influences within the womb, and it produces a wide variation as a function of forces, interuterine forces. And there certainly are forces that go on, as I said, up until the development of the adult brain. And you can now take any one of those adult brains, and in that adult brain, you're going to have a wide variety of possible outcomes, as I've indicated there. And as I will show at the end of my remarks, that in fact there are neuroscience advances which are going to take any individual brain and make a prediction about the outcome of that person in terms of things like reading, obesity, dyslexia, you name it. And the potential unintended implication of that technological advance is something we should think about. So all of this, of course, and this is the point, going back to the MacArthur thing, does any of this analysis, all of it interesting in its own right, does any of this analysis really impact the kind of inequality that is the central subject of this meeting? And I frankly don't know, and I don't think anybody knows. But it's important to know what's going on, and because one of these things could take off and have an implication, I think. So here's the 4 points I want to make. We're going to start off with what the machine is like when we arrive here. Is there a natural aversion to inequality? Do we come with that wiring from the factory, as it were? Do we have information that is the developing brain impacted by socioeconomic factors? There's work on that. Does variability in brain structure and function impact inequality? And finally, can neuroscience methods be used to predict social and cognitive outcomes? And will those further impact inequality? So let me try to cover those quickly and see where we go. Now, so the first one, is there a natural aversion to inequality? This is work that comes out of the field of developmental cognitive psychology. These people are so skilled at asking fundamental biologic questions with about 10 cents worth of supplies. This has always annoyed me, but they're incredibly adept at this. And so in this particular example from René Balgeron and the great psychologist David Premack, they were simply looking at whether children babies, in this case 12-month-old babies, care about the allocation of goods to toy giraffes, puppets that look animate. And so would the baby react to an unfair distribution of the food to the puppets? And the answer is yes. They seem to be very interested in a fair allocation of food. And if you make the puppets or the giraffes not puppets, but these rigid plastic ones, they could care less who gets how many bananas. So, so there's a sense that very early on, babies are they have an expectation for fairness. Now, this field, which is, as I said, rich, also captures William James' famous blooming, buzzing confusion that a baby must be experiencing, because there are other traits that these psychologists have depicted. For one, a reciprocity. So they can do these studies where A steals B's cookie, right? A steals B's cookie. Baby's not surprised at all if B steals it back. That just seems fair and square to a baby. And they have these ways of detecting whether the baby's happy or not. So there's reciprocity already understood on top of this fairness instinct. And here's another one I love. A goes in and hinders B. A goes over and hinders B. Now, B and C are part of a social group, right? So A hinders B, and then C goes over and helps A. What's the baby's reaction to that? Total surprise. How could that be? Don't you understand these basic relationships here? Because I do. I come with them, is the way this thinking goes. And we're retributive from the beginning. So A hits B, B hits A back. The baby's reaction to that, not at all interested. So that's all going on, and could that be leading to what happens then at the 5-year-old child? And new work, a lot of this work comes out of the way, by the way, from Paul Bloom and Karen Wynn at Yale. And they have a new series of studies out with Shaskin that are showing that, well, maybe there's a expectation of fairness actually when you give 5-year-olds the ability to choose whether to be fair, to distribute goods evenly. They actually prefer taking it all for themselves. And so there's this funny little time in our lives where maybe we started off concerned about fairness, but we settle into this take it for yourself, and then it all winds up with what is now the dominant thinking amongst the adults, which is that there is an inversion inequality. These are experiments done by the neuroeconomists. Who is it down there? I can't see it. The Caltech guys. Anyway. So in this kind of a simple experiment, they just take normal undergraduates and they give them a bag, and in the bag you either get money, or you start off with a lot of money, or you start off with no money, and then as they give the subjects additional money, they've determined that the people with money, in fact, are rewarded only if the goods are distributed to those without money, whereas those without money in the initial allocation, the reward mechanisms only light up if they actually acquire money. So one simple interpretation is, which we all know is crazy, that this would suggest those with money, read the Republican Party, would be glad to spread the future money around, and that doesn't seem to be the case. So there are these laboratory experiments we seemingly go quickly to wider implications, and I think a lot more work has to be done before we really nail down what the implication of this is. But then, are there basic brain mechanisms in this that seem to be calling upon altruism, empathy, concern about others versus concern about self? And there are intriguing studies. There are a lot of them. Here's just a couple I put side by side, where you take one structure in the brain that we know is involved in the processing of fear and all kinds of emotional relationships, the amygdala, and you look at the simple thickness of the amygdala in a variety of subject classes. And as you can see, as the amygdala goes down, this was a study on psychopaths, low psychopathy, high psychopathy. As you can see, as this structure becomes measurably smaller in the brain, there's a higher incidence of being high in the psychopathic measures that they have. And to the right is one that goes in the other direction. This is a study that just came out a few months ago in PNAS, and it's absolutely fascinating. They studied people who donate their kidneys to kin and compared people who donate their kidneys to total strangers. And one they're all kind of altruistic at one level. One's what they're calling low altruism, and one is called high altruism. And sure enough, it's detected by this. They detect these correlates with the size of the amygdala in the brain. So, is there going to be a science of looking at people's dispositions, being able to take a picture of your brain, figuring out what the dispositions are that are then weaved into a story about who we are and how that works, and that's another story. Okay, so concern about the self, concern about others might well have a biologic basis that is going to be played out and understanding it at a neural level. Is developing the brain impacted by socio-economic factors? This is work that started in a couple labs. This is the work of Bob Knight at the University of California at Berkeley. And he basically had been studying the effects of left frontal lobe lesions and had determined through a electrophysiological measure that people with left frontal lobe lesions reacted in a funny way as he measured it by doing these electrophysiologic recordings. And you can just see there in a little cartoon, you wire people up and you grab signals off their head and you give them this test where you show them a bunch of triangles and they're there to detect the one that's a little bit out of kilter. And that you get a brain wave associated with that. And then they throw in the surprise novel one there, as you see, of Mickey Mouse. And so people with left frontal lobe lesions respond different than normals, and it suggests some kind of left frontal lobe pathology if you get that kind of brain wave. So he does this experiment on low SES kids and high SES kids, and on just the simple detection test of the wacky triangle, everybody kind of behaves the same. But when you throw in the novel stimulus where you're supposed to ignore it and continue on the target task, the low SES kids begin to look like left frontal lobe patients, and the high SES kids look totally normal. So now he points out in his original paper that there's a lot of overlap, and this suggests to him that there's a likely, it's fixable if these children are identified and intervention studies are made that could turn this around. But it's a nibble. It's one of the earlier studies. This was done in 2009, I think. Early studies suggesting of a SES impact on actual brain systems. And then one just out last week that's been in the review process for a while by Nobla that came out in Nature Neuroscience takes a look at the association areas of the brain that we know are involved in higher level thinking and language and speech and other kinds of processing, and finding out that there is an increased cortical surface area as directly related to the level of education. So that in fact, this is another version of it from another study, but you have the finding that in fact, that expensive tuition bill you're paying for all your kids all those years pays off. And they are the fatheads you think they are sometimes, so there you go. So, but the caveat is that all these brain studies so far do not control for an important problem that the MacArthur Foundation and another group of work identified, which is are these absolute, in the SES context, are these absolute differences or are they relative? So the way the studies are framed now, they just have, in fact, identified that children that come from families or children who have low SES incomes, and they don't do the control where is it relative though? So a bus driver in one town and a bus driver in another town might be varying on the social status ladder, and it could be that the relative effect of where you are is making these differences in these brain measures. And if that turns out to be true, of course, that would be highly interesting in its own regard, but it would be a totally different interpretation than from the one that's sort of implied now. So I think these studies are fascinating and important, but they're young too, and they need lots of follow-up. Variability in brain structure, and I'm almost done here, so you can breathe easily. This is something I actually know something about. This is a cross-section of the brain, and that big white structure there is called the corpus callosum, and it's the neurons that interconnect your left hemisphere from your right hemisphere. And using these new high-powered technologies, you can look and see, well, gee, is everybody's brain wired the same way? And here are 4 individuals, just to give you a sense of it. And there are 3 areas of one hemisphere that were examined, the supramarginal gyrus, the angular gyrus, and so forth. So the red, green, and blue areas is all you need to know. And those are the 4 subjects that were being demonstrated here. And just looking at that, you can see literally that the fibers, volume, and actual placement from, if you combine the 2 pieces of information, are different in these 4 individuals. Now these are, look particularly at the one on the right, where you're looking at the projections of the supramarginal gyrus. Totally different parts of the callosum are receiving these fibers. Look at the ones where there's a huge number of fibers interconnecting the angular gyrus, the red one. This is the area associated with language and reading. Now, what does that significance imply? There's the physical evidence that there's variability. What does it imply for function? Variability in brain structure and function. These brain scans you see here are the typical new ones that you get. If you look at the images there on the left, they're showing the 19 neural circuits that are assumed to exist now. If you group together, you put somebody in an MR machine and you measure all the activity that's going on, and you correlate the activity, says what are the circuits that are firing together, and you map those all out. Well, they figured out now there's about 19 of these circuits. That's the current dogma. And then you ask yourself, well, how much variation is there in those 19 circuits from person to person? And as you can see from the lower picture there, the variation occurs in the exact areas where you would have higher order thinking. You would be attributing higher order thinking. So the variation in the size of the area, as indicated, the higher it is, it gets yellow, is right where you would want it, you would think, for explaining the variation in cognition and other kinds of behavioral traits. And the blue areas, which doesn't show much variation at all, is a part of the cortex that is involved with motor activity, and the other parts are the sensory areas. They're not going to change much. So you get all the very inter-individual variability in the associational parts of the cortex, and that's just more of the same. And so another thing people don't realize, the big picture there of the brain activation, you see a nice picture of people show you, well, this is what the brain looks like when you do X, right? Well, actually, that's an average of what the brain looks like. Those are all the brains that went into that study. And if you take the you take a person and you get their brain distribution to a task, and you bring them back into the lab 6 months later, you can see there's a nice correlation with how their brain responds. And those are 2 subjects doing the same thing. Whereas there's low correlation about how they are between themselves. So we seem to have our set individual natures pretty much set, and it's consistent through time. So this is the final slide then about the double-edged results, the double-edged nature, the dual use nature. So this is a paper that just came out by John Gabrielli at MIT, where he summarized a body of research that is predicting, as I said at the beginning, predicting the outcome of people. So there's one, the first one there is predicting, I can't see anymore. Yeah, the reading outcome. So imagine, so there is now a technology. You could take your child into the MIT scanner. They'll do a brain scan on you. And let's say the child had dyslexia, and you want to see if there's any chance this training that you're expensively committed to is going to pay off with increased reading scores. And the answer is yes. They can predict that now because of the clever way they've set up these studies. The one below that has to do with recidivism. There's a big problem in prisons. Who do we let out? Two people come up to the parole board and they can't figure out, should we let Jones out? Should we not let Jones out? What's the deal? And they talk around the table with each other. And if you really talk to these experts who do this all day long, they say, give us some data. How do we know which one to let out, right? Well, so here comes brain imaging along, and this is one of the studies that came out of that law and neuroscience project. And it turns out you can predict the extent of recidivism, the rearrest as it's put in the slide, by looking at the actual brain imaging and doing analysis. And another one is on addiction, and another one is on eating. So this is a quick survey. My intent is to show you there's lots going on. My intent also is to say that I think the field is young, and I think it needs a lot of shaking out. But I think it's something that both economists and those interested in the larger social questions and the neuroscientists themselves should continue this interaction to shake these things out. So, thank you.
All right, so we have plenty of time, half an hour for questions and comments and so on. So please address your question to either or both of the speakers. And do you want to take a bunch, or do you want to take them one by one? Okay, we'll see once the questions are asked. So, any questions? Yes.
Hi, I'm Els Torello, Open Society Foundation's Public Health Program. Thank you for the very interesting interventions. I was struck a little bit, I'm a biologist myself, and the discussion about genetic causality and linked to that genetic determinism has been very active already for many years in biological sciences, medical sciences, etc. And it's interesting to see how you actually take that one step further into behavioral threats, whether yes or no. And I think you made Daniel, you made the point very clearly that if there is anything, it's probably very small, the influences. And yesterday we heard from Thomas Piketty, we saw this amazing graph showing that there is a very direct correlation between the level of income of parents and the level of higher education that kids have. Like a very strong correlation if you compare that with the very weak and probably multi-snippets type of interactions we may see with behavioral threats. Aren't you afraid that we feed the discussion about genetic determinism as an ideology, which, as you are probably well aware, has been exposed already, for instance, by Richard Lewontin in his book like 20 years ago, Biology as an Ideology, and that we actually come to conclusions that entrench inequality as a result of biological whatever determination. It's all in our genes, and even if you make a very nuanced case that that's not what we need to see in or read in that, it's often what people take away, and I wanted to have your thoughts on it, more from a macro perspective, seeing that the data are really there, that there may be something, but how that then is interpreted into big conclusions that may not warrant and that are actually a little bit dangerous.
So my view is that we, as scientists and as educated citizens, we should try to be accurate in our understanding of the way the world works. And I think that that what we're seeing, I mean, I think that the kind of evidence that Thomas Piketty showed yesterday is consistent with either strong environmental effects or strong genetic effects. The direct evidence from the heritability studies is that the genetic effects are, in the range of 40% for things like education or income taken all together. And then I think we need to couple that scientific understanding with also an understanding of what that does and does not imply about political conclusions. And I think genetic determinism is simply fallacious from a logical point of view. It just doesn't follow from the scientific evidence on heritability, and that is just as important a message as the scientific facts. I think it's important to help people understand that even if genes play a role, that doesn't in any way reduce the role of environment or reduce the role of policy in combating inequalities in society.
Yes, I think you answered the question. Everything has dual use, and you want a culture that just knows that, and they're grown-ups. And you could say that half the problems of the world today are from cultures who don't accept the fact that there's a larger informational pool you have to understand before you decide on the nature of something. And I can't imagine that science is supposed to start buffering what it learns or how to think about what's the next proper thing to learn by worrying about people misunderstanding what may come out of it. It starts getting into all kinds of problems. This came up when I was a version of this story came up when I was on the Bioethics Commission, and we tackled the problem of stem cells, and the stem cell issue brings up the problem of the moral status of the embryo, and as everybody knows, that is a hot potato issue in any culture, especially the American culture. And when there was a full discussion of the issue on a committee of 18 people, a good half of which prior to the discussion were just reflexively against the idea of biomedical cloning because it would tamper with a blastocyst. By the end of a 6-month discussion and scientific presentations and all the rest of it, the majority vote was that it was morally okay. Now, and this was under full public disclosure, and so the conversations and the debate and the discussions, you got to believe that it ultimately works, or you're not living in a good culture. So.
Other questions? Yes.
Connell Smith, OECD. So 2 questions. I have actually one at each. For Daniel, the idea of using, as I understand it, an index of the combined genetic effects as a control. My gut reaction is, when I think about this, is it's doing one of 2 things. One possibility is that part of the causal pathways you're trying to understand go via some of the genetic stuff that you're controlling for. So you're actually the control is actually obscuring what you want to know. The other possibility would seem to be they don't, in which case what does the control add? What do you actually see substantively getting out of having better controls done in this way? And for Michael, this is much less in depth, but I remember looking at sort of law in neuroeconomics 10 years ago, and there was a concern with sort of people saying, well, law has a distinction between actions undertaken by someone who is neurotypical and the defence of mentally unwell. My understanding is a lot of the neuroscience suggests that it's very difficult to draw a biophysical boundary between these two states. You know, to what extent does this end in my brain made me do it as a defence?
So the question was, if you use a polygenic score, an index of genetic variance, as a control variable, then how can that help? Either maybe that the effect you're interested in is mediated by the genes, in which case controlling for them wipes out the effect, or it's I forget the other possibility, but yeah. So the answer is that I constructed my example very carefully because I proposed a randomized experiment where we're looking at an intervention like we're randomly assigning half of the children free preschool and half not, and then we're looking at the effects on later life educational attainment. And by construction, when you do a randomized experiment, it's uncorrelated with anything else that you might control for. So it's not correlated with the polygenic score. And therefore, you're going to get an unbiased estimate of the effect of the intervention, no matter what you control for. The only effect of including control variables is to absorb residual variance and reduce standard errors, increase the precision of your effect estimate. So I'm quite confident that that's going to be, within the next few years, a widespread use of polygenic scores, because we'll have polygenic scores for many kinds of outcomes, and this kind of use of them will be possible and very beneficial in social science. But the issues you raise become important and serious issues if we think about non-experimental studies where you're trying to estimate effects. And then there are all sorts of tricky issues, and you have to worry about exactly the kinds of exogeneity issues and over-controlling issues that often come up in econometric work.
Well, the short answer is that one of the lessons learned is that the neuro defense as an excuse doesn't go very far. Not only are most studies averages, and how does that apply to Mr. Jones sitting there in the defendant's seat? But also that the realization that it came out of many of the discussions of this project, that a typical defense now is called the left frontal lobe defense. That's disinhibitory with respect to violence, and if you have it, therefore you have an excuse, and blah, blah, blah. Well, it turns out, if you actually look at the data, there's about a 3% baseline rate of violence, and if you have a left frontal lobe lesion, it jumps to 13%. Well, that's 87% of people with left frontal lobe lesion don't go out and cause harm. So that kind of data made people very sober about it. But my own personal view is that you don't look for responsibility in the brain. The brain is at the social level that we all live in. And the minute there's more than one person in the world, you make a contract with them. You just say, okay, here's the deal. And that's what we're all part of. And so if you violate a social norm, you will have to be held accountable for it at the social level. Now, what you want to do with that person is a separate kind of political social thing. You want to be retributive, you want to be not retributive, you just want to get them off the island. Those conversations are long and deep. I don't know why. One of the thoughts I had after working with the legal system, I don't know why the sentencing phase of a trial has to go to the legal system. I don't know why professionals aren't brought in who deal with these kinds of issues, aren't deciding what's the best treatment. But that's probability of that happening is close to zero.
Further questions? Yes.
At what point, if there's let's say that, because the discussion kind of is involving inequality, and at what point does the fact that society, the inequality in society, make the data collection rather than the actual methodology that you're developing impossible to actually do any real kind of research with it? Because if you're talking about heredity in the sense of accumulation of income, let's say, and you have like people who are rich, the kids of those people are inheriting that richness regardless if they collect the good behavior traits or not. It almost ruins the randomness of the sample. Or if you have, you're talking about like brain structure as something that's, you're going to take a look at like the frontal lobe, whether or not that's going to lead to recidivism of criminal behavior. But there's populations that are being targeted that may not even be exposed, like they may not be even real criminals in that sense anyway. It might be just like certain groups are being targeted for like where they live or what their income is in general. Is there a point that you almost can't take anything out of it other than the methodology, or is there always a way to take something meaningful out of it?
You keep hammering away at it. These things won't be resolved next Tuesday. And I think at some point, some of these things will become clear. There's nothing you can do, or there's things we can do where interventions are important, or whatever the issue is. I do think that it is extremely important that these, let me tell you something. When I went on the Bioethics Council, I thought it was ridiculous. And then I started thinking about all the questions that come up, questions you're referring to. They're not ridiculous. They're things to be thinking about because the speed of scientific discovery is of such a nature, these things are on your plate before you know it. And if you haven't thought about it, you're just behind the game. So I'm all for these kinds of discussions, these kinds of almost gaming the future to get ready for what I think will inevitably be vast scientific discoveries. So anyway, you just keep going on it, and you see when it comes down the pike what it'll mean for your social culture. I don't know if that's an answer or not, but you tried.
I mean, I just think that the ethical issues and the policy issues are extremely important, and the kind of work that the Bioethics Commission does and is extremely important, and we should be thinking about what the downstream ethical and policy implications of this work will be. I think it's part of our responsibility as scientists, and I think that we as a society need to be prepared.
Research. So my brother-in-law is a social scientist, and occasionally he'll do some research that doesn't really match his progressive values politically. I'm just curious, what have you ever had the experience of, or how do you think about morally and ethically and personally when your research findings start to hint at things that frankly you disagree with, find anathema, would have concerns about if you released it? For instance, what if you'd found things in this research that suggested to you things that we simply just don't want to accept, couldn't accept, and would refuse to accept? Did the question make sense at least? Okay, let's check.
So I think, fortunately, I don't think, I've never been in the situation that you're describing. I tend to think that that knowledge in itself is morally neutral, and that it can be used for good or bad purposes, and that as scientists we have a responsibility to report what we find, even if it goes against what we expected or wanted to see. And that actually is a moral obligation on us as scientists. but I've never been tested in the way that I think you're imagining, and I'm not sure how I would react if I were.
So they do studies on differences between men and women on how they react to disconfirming data. And men basically want it out of their room, and women are more accepting of it and trying to deal with it. This is work done at Montreal by Kevin Dunbar many years ago. And so there are certainly differences that people can, or that do occur. And I certainly know of a case exactly as you posed, that person chose not to pursue it because of the possible misunderstanding with social implications. So he just didn't want any part of it. But there was something else you said. I'm just losing what I had a thought on it, but I'll just leave it at that. So.
Actually, can I add one more? Yeah. Just one more thought on what my group, the Social Science Genetic Association Consortium, has done along the lines of what you're asking. You know, because there are many ways in which genetic results can be misinterpreted, and often are, we've adopted as a policy to release alongside any major paper a frequently asked questions document that explains to an educated lay audience what we did and what we found and what it means, and then also what it doesn't mean and what conclusions you shouldn't draw from the work. And our policy is to, as much as we can, if the journal allows it, post it on the journal website or post it on our website along with the article, and also distribute it to any journalists who contact us about the work, to try to play our part in preventing misinterpretation of the findings.
So an unrelated question to the last one. Genetics matter for social and behavioural outcomes. A big sample size is also really important, and I think this probably stretches beyond genetics. There are a lot of small effect sizes in the behavioural social sciences. What are the implications of this for the statistical infrastructure we want to have at our disposal for economists over the next couple of decades? What should we be trying to build? When you talk to national statistical officers, they refer back to the NSO for their system of national accounts, for their model of a statistical infrastructure, which is not, I would argue, just not relevant to the kind of questions we're addressing. If you were building a system of social statistics, taking into account what you know about causality and effect sizes, what would it look like for a country?
So I haven't thought about this at all, so this is shooting from the hip. But I think that if we're studying phenomena where we expect the effect sizes to be small, we know we need enormous sample sizes and stringent significance thresholds if we're going to have credible results. So the statistical infrastructure would have to, the data infrastructure would have to take that into account. We also know that in these social, when we want to study economic or social science questions, exogenous variation is very hard to come by. So we'd also want to design the data infrastructure in a way that that maximizes the chance that we can get that kind of variation in the data. In the U.S., that presumably means you want to at least collect data across many states because you have little natural experiments going on in the way different states do things. But there may be other ways that you can also try to introduce exogenous variation, maybe in question order, if you want to study the effects of the survey itself, for example.